We work in the field of catalysis, and for catalyst researchers, such methods as scanning tunneling microscopy, optical microscopy, scanning electron microscopy, transmission electron microscopy are very common. And for catalytic materials, also very common to have appearance like you see at the slide. And typically you have several tens, hundreds, or even thousands of objects on one image. And the task is to count these objects and to find average diameter, some characteristics of shape, and so on. One more complication is that typical series of images may contain 1, 2, 3, 10 images, and then you work with another sample, and it means that the model you trained on these images may be not applicable to another series of experiments. Researchers, when we try to use a typical approach to annotate full image, several hundreds of thousands of samples of objects, it was very time consuming, and researchers asked to do something more simple, more user friendly. We tried to use few shot learning on this kind of images, and in order to this specific case to be distinguishable from other cases, we suggest to call it subshot learning. It means that you have homogeneous image which is made of essentially similar parts, and you have part of this image crop and annotate all the object inside. Then you use this crop to train the neural network, and this trained network may do inference at the same image or on another image in the same series. This is our configuration, experimental settings. We used Cascade Masker CNN with ResNext 101 backbone, which was pre-trained on Microsoft Cocoa dataset. During training time, we used crops of 64, 128, 256, and modest augmentation, which was mainly flipping, color normalization, and 3 learning rates in our learning scheduler. During test time, we applied original image size, no resize, and also normalized color to these values. This approach was tested on a custom dataset, which was collected from scanning tunneling microscopy images. Our set contained 8 images with about 1,200 of objects annotated, and test dataset collected contained about 700 of annotated objects. So you see here comparison of results for network trained on the complete data set of 8 images. Then step by step we trained this on first, second, and so on images, and we see that this network trained on only one image shows much worse performance, less metrics than trained on the whole data set. If we train it on a crop of smaller size, then we see further decrease in performance. However, sometimes it's not so. So we see, for example, for image number 6, we see the increase in performance instead of the drop. And it becomes evident that choice of crop is very important here. And we see that when we compare crops, training on the crops chosen by operator and crops chosen randomly, we see systematically better results after training on crops chosen by operator. Even more, if we let the operator to use crops of higher size, 1/2 of the image, the crops can even outperform the full data set. So whereas, for example, 2 orders of magnitude more annotated objects than on this crop. And the choice of crop seems to be very important. We see that if we take random crops of different size, we see very big scattering, very large scattering of the metrics, and sometimes very nice crops can compete with much higher. For example, here in this experiment, crop of size 64 was performing better than any crops of larger size. In fact, we cannot say right now why the rule of thumb how to choose the crop. However, we implemented this service as a no-code service for researchers where they can annotate using graphical annotating tool, label me, annotate it on the desktop, then send a message to messenger, and after that, bot trains the neural network and produces results on the same image. And also you can use this trained network in further experiments and to test this network on images of the same series. And here you can see one of the typical results when in 10 minutes using this subshot learning approach, about 2,000 of objects were obtained, and some statistics can be automatically processed and obtained for these experiments. Thank you very much. Would be glad to hear your questions.
Yeah, sorry for this. My audio had some issues. Yeah, so if we have time for a question, so if somebody would like to ask any question, please ask Alexei directly. So I have a question, Alexei. So can you describe what is the biggest challenge in your approach that you're facing?
The biggest challenge is the biggest challenge for researcher is to take right crop. As I told, we see that researchers perform much better than just random cropping of the image and taking the part of the image which is the most representative. So human is better than artificial intelligence in this sense. However, we cannot right now understand why and which rules use humans, which part of the image is more representative and should be used for training and which are not.
Yeah, it's a good point. Okay, well, thank you very much. And now we're moving to the next talk, titled Unsupervised Artifact Removal in Histopathology Images by Joseph Boyd from Université Paris-Saclay. So,
yes.
Yes. Just say.
Am I audible? Hello?
Yeah.
You can hear me, okay.
Yeah, I can hear you, but we need to share your screen.
Yes. Sorry about this.
Okay, now it's okay. Yeah. Oops. Can you hear us?
I'm sorry.
Okay.
You can hear me.
Right.
Sorry about that.
Okay, now you can start. Yeah.
Thank you. Yes, sorry. Yes, good evening. My name is Joseph Boyd. I'm a postdoc at the Mix Lab at Centrale Superdec here in Paris. So good evening from Paris. This is a work in progress presentation titled Unsupervised Artifact Removal in Histopathology Images. So let's jump in. So there's an abundance and a diverse range of artifacts, image artifacts, which appear in whole slide histopathology images. So we've identified 7 categories of these shown here. And as you can see, they range from anything from foreign objects appearing in the image field to morphological changes such as tissue folds. So there's a wide range of these things. And so we compiled the data set from 28 whole slide images harvested from the TCGA database, which is currently missing in action. If anyone knows what happened to TCGA, please let me know. From these whole slide images, we extracted 6,500 or so non-overlapping tiles at 2 different magnifications, half of which contain one of the 7 artifact types that I indicated, half containing just regular tissue. So we have some sort of multi-domain unsupervised problem. And the baseline model that we selected for this problem, for artifact removal, was the cyclogan, which we've already seen today. But I will just recap some basic points. So the cyclogan is an unsupervised model for unpaired image-to-image translation. We have 2 discriminators, each mapping from one domain to the other. And they have already shown various successes, including the one we've seen today. There's a very nice review from Trushnig, Gademeyer from 2020, showing the applications of cyclogans to histopathology images. So a little more formally, we have these 2 domains, the domain A of artifacted images and the domain B of clean images, clean tiles. And then we have, in the usual way, 2 generators. The important one being highlighted here in red, the AB generator, that is to say the generator mapping from the artifacted domain to the clean domain. That is, in essence, the artifact removal system. So some details. We implement attention unit generators and self-attention patchcan discriminators, more or less standard. Due to the relative shortage of data, we've incorporated this recent adaptive discriminator augmentation, ADA, from Keras, which is a sort of online, it's a way of prepending differentiable augmentation operators to the discriminators themselves. And this had a particular significant impact on the overall results. Then we run a 80-20 train test split, and then we evaluate at a fresh A inception distance of 50.28, which may seem relatively large, but it was evaluated on a relatively small number of images. And for some comparison, some perspective, the FID between the 2 domains of the test images from the 2 domains is 84.17. So we're making some kind of improvement. And so what this looks like here is some samples from the artifact domain in the left of each of these panes. And on the right, we see the model output, the AB generator model output, and we see that it's relatively successful in cleaning these artifacted tiles, even in the challenging case of the tissue fold in figure D, where the morphology itself has been changed. So we have some success. And more recently, we've been thinking about how to extend this model from an unsupervised setting to a weakly supervised setting. And we've done this by proposing the following extension to the cyclogan, specifically to the AB generator of the cyclogan. And that is to append a attention network to the penultimate layer of the generator, which produces a attention map, alpha, which is used to reweight the generator output and with the input. so as to ideally decompose the artifacted image into the foreground and the background, the artifact and the non-artifacted region. And we support this with a weak supervision. So we annotate the artifacted images into the 7 categories we can consider to be a weak label. So there's no segmentation, no manually annotated annotation of segmentation. And then we guide the attention with an additional auxiliary classifier C, which receives as input the pixel-wise product of this attention map and the artifacted image. That is to say that it sees the attended regions of the artifacted image. And we finally regularize this attention map with sparsity and smoothness priors. And we firstly improve on our baseline with an improved fresh area inception distance of 49.81. We report also a classification performance of this classifier of 92%. I should emphasize here that this whole thing is trained end to end. And then we see in the figures, we see in the top row examples of artifacted images, inputs to this AB generator, this extended AB generator. We see in the center row the attention maps, where it's localizing the artifacted regions. And in the bottom row, we see the cleaned output. There are a number of advantages to this so-called copy-paste attention. One is that it appears to facilitate the artifact removal. Secondly, it provides a sort of bonus auxiliary output in the form of this soft segmentation, this attention map. And finally, and importantly, it more or less locks the background regions to the input values. That is an important property of a artifact removal system, in that you don't want to modify the any part of the image which is not an artifact. So this shows some promise as a work in progress. Now I'll close with some perspectives. First of all, on the data side, we saw that this dynamic ADA data augmentation had a significant impact on the performance. So what could be gained, for example, by doubling the available data, which is relatively easy to do in a weekly for a weekly supervised data set, provided our esteemed TCGA makes a comeback. The next question that we're interested in is how to resolve this tension between the adversarial training and the other auxiliary tasks, because there's a certain tension between them. And it actually brings us to a final question, which we intend to endeavor to answer. And that is, do we actually need the full cycle GAN anymore? Or have we now outgrown it with this weak supervision? So that's the, yes, thank you. That's the end, and open to questions.
Okay, thank you very much. One question in the interest of time. You can ask directly, Joe. Yeah. Maybe I can ask you a quick question. So what's the horizon of your work? Like how it's in progress, right? Your work. So how much time do you think you will be able to achieve these goals?
Well, I mean, it depends on the state of availability on the one hand. But I mean, we have many ideas to extend this, and I think it's a question of just running these experiments and seeing what comes out of it. So I would say weeks to months, okay. Maybe the summer,
Yeah, these GAN methods will work well if you have data, enough data.
For sure, yeah.
Okay, well, thanks a lot for your talk, and
thank you very much.
Interesting direction. So I'll give the floor now to Dan to announce the next speaker.
Great. Thanks everyone for the great talks. It's kind of fun to see these strings that do connect presentation to presentation with respect to synthetic data generation, simulation, histological analysis, and it's always a mixed bag at the beginning of this meeting, but it's kind of fun to see the classifications of different sorts of strategies emerging as competitive, and it would be kind of fun to see who wins at the end with respect to favorite application for segmentation and classification. Our next speaker is Dr. Kristen Maynard. Are you there? Oh, you are there. Excellent. So Dr. Maynard is a research scientist at the Lieber Institute for Brain Development in Baltimore. It's part of Johns Hopkins Medical Center in East Baltimore. Her work historically is focused on connecting neural cellular function to brain network function, and finally to behavioral outcomes, the idea of sort of cells to circuits to behavior. And she's generated really influential studies that demonstrate how the contribution of individual cell types can, in fact, influence behavior. More recently, Kristen's focused on going backward from behavior to cellular phenotypes, starting with behavioral disorders such as schizophrenia, then applying cell type specific labeling methods and detailed gene expression analysis to understand how the molecular effectors of genetic risk for schizophrenia across spatial fields of the brain. And it's the combination of kind of unique labeling, microscopy, and the use of spatial data to understand disease mechanisms that compelled the steering committee to invite Kristen to join us today. So we really look forward to your talk, and thanks for making time with this group that's very much outside of your comfort zone, but I hope you get a benefit from the follow-ups.
Good afternoon. Thank you, Dan, for the kind introduction, and thanks to the organizers for the invitation to speak at this exciting workshop. So as a molecular neurobiologist and microscopist, my goal today is to give you all kind of a comprehensive introduction to the spatial transcriptomics work that we've been doing to better understand the topography of gene expression in the human brain. Given that spatial transcriptomics is sort of a nascent field with new technologies emerging almost weekly now, there's lots of opportunities for tool development on the computational side, especially in terms of image analysis. So I want to introduce a bit of what we've done, what others have done with our publicly available data, and where we hope to go in the future. So a remarkable aspect of the human brain is its highly organized structure-function relationships, especially in the cerebral cortex. So the cortex plays a key role in many functions, including perception, awareness, thought, memory, language, and movement, to name a few. And specialized cortical areas subserve each of these functions. The dorsolateral prefrontal cortex, which I'll be referring to as the DLPFC, is especially important for executive functions, including working memory, cognitive flexibility, and planning. And disruptions of this brain region have been associated with several psychiatric and neurodevelopmental disorders, including schizophrenia and autism spectrum disorder. The spatial organization of the neocortex reflects its complex function. So a defining architectural feature of the cortex is its subdivision into 6 layers, each containing a unique distribution of different cell types and connections with other cortical or subcortical brain regions. In the cortex especially, the position of a cell within a specific layer influences its gene expression, its morphology, its physiology, and its function. And indeed, studies in the postmortem brains of individuals with schizophrenia and autism spectrum disorder have implicated not only specific cell types in these conditions, but also revealed differences in neuronal and synaptic structure that are spatially localized to specific cortical layers. So one of the ways that we can understand the functions of different cell types in the brain is to study what genes they're differentially expressing. And in the past few years, there have been several rapidly evolving transcriptomic approaches for studying gene expression in the brain using RNA sequencing. I kind of like this Lego brain schematic, which illustrates how we've expanded from bulk RNA sequencing, which contains a mixture of cell types, to single cell or single nucleus RNA sequencing, which allows us to understand molecular profiles for different cell types. And then most recently to spatial transcriptomics, which aims to link gene expression to its anatomical location to add molecular insights into structure-function relationships. And in fact, spatially resolved transcriptomics is a rapidly evolving field that was actually just named Method of the Year by Nature Methods for its power to unlock relationships between gene expression and the spatial organization of complex tissues. So to further our understanding of gene expression within the context of the spatial organization of the human cortex, we use the recently released 10x Genomics Visium platform to generate spatial maps of gene expression in the 6-layered dorsolateral prefrontal cortex of the adult human brain. And for those of you who aren't familiar, Visium uses a patterned array of spots with barcoded oligo-DT primers to capture the mRNA from a tissue section. And then during cDNA synthesis, which occurs on the surface of the slide, barcodes indicating the location of each spot are incorporated into the cDNA, which is then removed from the slide, prepared into libraries, and sequenced. So this basically allows each read to be mapped back to the correct spatial coordinates on a histological image. And resolution here is limited only by the spot size and the spacing on the array. And in the current platform, each spot is about 55 microns in diameter, and they're spaced about 100 microns center to center in a honeycomb pattern. We also use the 10x Genomics single-cell RNA sequencing technology to identify molecularly defined cell types in human DLPFC. This sort of droplet-based approach uses uniquely barcoded gel beads, which are mixed with nuclei and then added to an oil surfactant solution to create these single-cell gel beads and emulsions, or GEMs, which act as individual reaction vesicles in which the cDNA for that nucleus is barcoded. And this takes place on a piece of equipment called the chromium controller. These barcoded products are then pooled for downstream reactions to create sequencing libraries. And after sequencing, the unique barcodes are used to map the reads back to their original single nucleus of origin. And I just want to point out that while single-cell RNA-seq approaches are common in rodent brain tissue and cultured cells, the fact that most available postmortem human brain tissue is frozen has really resulted in the majority of data in the human brain being generated on isolated nuclei with single nuclei RNA-seq approaches. So to give a brief overview of the talk, I'll be describing how we use Visium to identify layer-enriched genes in the human cortex and how we validated these findings using in situ hybridization. I'll also talk about spatial registration of single nucleus RNA sequencing data, unsupervised data-driven approaches for analyzing spatial gene expression, and the power of spatial transcriptomics to better understand complex brain disorders. And on the way, I'll be highlighting how we and others have leveraged the microscopy data that accompanies Visium gene expression to better understand spatial topography in the brain. And I'll try and discuss opportunities for further integrating these different data modalities. So before I jump into the science, I want to highlight the work I'll be presenting as a collaborative effort. The wet bench data collection for this project was led by myself and Carrie Martinowicz in collaboration with 10x Genomics. And the data analysis was led by Leo Collado-Torres and Andrew Jaffe in collaboration with Stephanie Hicks and Lucas Weber at Johns Hopkins. So in our study, we profiled human postmortem DLPFC tissue sections from 2 pairs of spatial replicates from 3 independent neurotypical adult donors. So each pair consisted of 2 directly adjacent 10-micron serial tissue sections, with the second pair located about 300 microns posterior from the first, so resulting in a total of 12 samples run on the Visium platform. We acquired the tissue sections in the plane perpendicular to the PL surface that extended to the gray-white matter junction, and we included the 6 cortical layers as well as some white matter. And we sequenced each sample to a median depth of about 300 million reads, which corresponded to a mean of about 3,500 unique molecular indices and about 1,700 genes per spot. So the orientation of each sample was confirmed using marker genes for the gray matter containing neurons, the white matter containing glial cells, and then previously identified laminar marker genes. So what you're looking at on the top panel is a DLPFC tissue block and corresponding histology from a representative section. The bottom panel shows spot plots depicting the log transform normalized expression, or log counts, for SNAP25, MOBP, and PCP4, Expression of these genes confirm the spatial orientation of each sample by delineating the border between the gray matter marked by SNAP25, which is enriched in neurons, and then the white matter marked by MOBP, or myelin-associated oligodendrocyte basic protein, which is enriched in oligodendrocytes, and also defining layer 5, which is marked by PCP4, a protein that's important for synaptic plasticity. So to give you a flavor of the collective data set, here are the spot plots for one gene, PCP4, for all 12 tissue sections. So there's one row per subject, and adjacent spatial replicates are shown in the columns. So you can see there are slight differences in the dissections for each subject due to the folded nature of the cortex in the human brain. But we have the span of the cortical layers in the white matter with cortical layer 5 enriched in PCP4. So using these data, we first generated aggregated layer-enriched expression profiles for each spatial replicate using a supervised approach. So we used cytotectonic architecture and robustly expressed region or layer-enriched markers combined with a dimensionality reduction method, specifically T-distributed stochastic neighbor embedding, or T-SNE, to assign individual spots to each of the 6 neocortical layers or the white matter. Then we performed pseudo-bulking by basically summing the UMI counts for each gene within each layer across each spatial replicate to generate layer-enriched expression profiles. The pseudo-bulking approach summarized approximately 48,000 spots to 76 layer aggregated profiles across the 12 samples, thereby removing sparsity and greatly increasing the UMI coverage of the genes. Principal component analysis of the layer level or pseudo-bulked expression profiles across all sections and subjects revealed that the first principal component separates the white and the gray matter, and the second principal component associates with the different cortical layers. So after pseudo-bulking, we used 3 strategies to perform differential expression analyses using these layer-enriched expression profiles with linear mixed effects modeling. The first statistical model, termed the ANOVA model, involved testing for differences in mean expression across the 6 layers plus the white matter and asked, is any layer different? The second statistical model, termed the enrichment model, tested for differences in expression between one layer versus all the other layers and asked, is one layer greater than the rest? And then the third statistical model, termed the pairwise model, tested for genes differentially expressed between each pair of layers, so 21 pairs, and asked, is layer X greater than layer Y? So you can see the visual depictions of these 3 models using MOBP as an example. And remember, MOBP is a gene highly expressed in oligodendrocytes, which are enriched in the white matter. So using this approach, we systematically assessed the robustness of previously identified layer marker genes from rodent and human studies in our DLPFC layer-enriched gene expression dataset. We found that a subset of these canonical marker genes from the literature, so about 60% of them, showed significant differential expression in our human DLPFC data, which were largely driven by markers identified in the human cortex as opposed to the rodent cortex. We also identified several genes previously underappreciated as laminar markers in the human DLPFC. So here are just a few example genes, including AQP4, which is enriched in layer 1, HPCal1 enriched in layer 2, TRABD2A, which was enriched in layer 5, and then KRT17, which was enriched in layer 6. So the box plots and corresponding spot plots for these genes are depicted on the left. And then we validated these novel layer-enriched differentially expressed genes using multiplex single molecule fluorescent in situ hybridization, or SM-FISH. So you can see AQP4 in red, HPCal1 in white, TRABD2A in pink, and KRT17 in green, with lipofuscin tissue autofluorescence segregated into its own channel. And I'll just briefly highlight that we've developed some now published software called ... for quantifying single gene transcripts in individual cells and predicting cell types from SM-FISH data. We're currently working on higher plex assays and automated imaging capabilities to be able to visualize more genes across larger areas of the brain, which will be important for validating and adding single cell resolution to genes of interest that come out of these kinds of Visium studies. And as you know, and we've kind of been talking about today, segmentation of nuclei, cell bodies, and individual gene transcripts in complex tissues like the postmortem human brain is really a significant computational challenge on the image analysis side, especially because postmortem human brain has high autofluorescence due to lipofuscin, which is a product of lysosomal digestion that increases with age. So as multiplexing increases, this is an obvious area where computer vision approaches could make strides in the analysis of SM fish data, especially highly multiplexed data across large areas of tissue. So beyond analyzing and validating Visium gene expression data, we've also performed cell segmentation of the accompanying histology images to estimate the number of cell bodies located in each spot. On the left, you can see an example of this segmentation in mouse brain, which has a much higher density of cells in the cortex compared to human tissue, which is depicted on the right. On average, in our human tissue, we had about 3 cells per spot, with about 15% of spots per sample containing a single cell body, and about 10% of spots containing no cell bodies. And we actually consider these spots lacking cell bodies to be quite interesting. These kind of neuropil spots are enriched in the dendrites and axons that form the connections or synapses between neurons. And indeed, using a previously published data set of genes expressed at rodent excitatory synapses, we identified significant enrichment of genes that are preferentially expressed in the transcriptome of synaptic terminals in these neuropil spots. And this is really particularly relevant for brain disorders, as it's been shown that genes associated with increased risk for schizophrenia are preferentially enriched for synaptic neuropil. So suggesting that this sort of extra nuclear information that's captured by Visium, but missed by single nucleus RNA-seq approaches, may really be especially relevant for understanding genetic risk for brain disorders. So we're very interested in leveraging this histology image data and quantifying other parameters such as cell size, shape, or other unbiased features or patterns that may not be biologically obvious for integration with gene expression data. And again, this is another opportunity where machine learning approaches may be able to extract new types of meaningful information from the images that we can then overlay or incorporate with cell and region-specific gene expression to build more integrated anatomical and molecular atlases. I also want to briefly mention that while most of the data I'm talking about today is focused on laminar organization of cortical tissue, we're also expanding into other brain regions, such as the hippocampus, with more spatially complex circuitry and morphology. Stephanie Page, director of our imaging core facility at the Lieber Institute, has been optimizing Visium data generation in this hippocampal region, which is quite large in the human brain and will require the use of multiple Visium arrays to fully cover the structure. An analysis of larger and more anatomically complex brain regions will require additional computational strategies on both the image analysis and the gene expression sides to be able to kind of stitch together multiple arrays and reconstruct a lot of these subfields. And you can imagine doing this across different donors with slightly different dissections also poses significant challenges. So I want to move now from comparing gene expression across cortical layers to integration of Visium data with single nuclei RNA sequencing data. There's really a growing interest in combining these data modalities and a number of tools coming online to support these approaches. So here's a schematic of what I mean by spatial registration, which is essentially taking clusters of cells or nuclei derived from single nucleus RNA-seq data and mapping them onto the different cortical layers. So to validate this approach, we first use single nucleus RNA-seq data from the Allen Brain Institute, where they manually dissected each individual layer of the medial temporal gyrus of the cortex, and then performed single nucleus RNA sequencing and clustering of laminar cell types. The heat map shows the correlation between enrichment statistics computed on the single nucleus RNA-seq data on the x-axis against those from our laminar Visium data on the y-axis. And if you focus on the dark green squares showing strong correlations, you can see that the Allen brain cell types manually dissected from each layer nicely map onto the cortical layers annotated in our Visium data. This validates the Visium platform and also demonstrates the robustness of this kind of spatial registration approach. So we also wanted to demonstrate the utility of spatial registration for data sets where laminar information is not known a priori. So we generated our own single nucleus RNA-seq data from bulk DLPFC from 2 individual neurotypical donors. This work was done in collaboration with Matt Tran, a graduate student in the Martinowicz lab. And we identified about 6 broad cell types across 5,200 nuclei, including both neuronal and glial populations. We did further clustering of these populations, and on the right, I'm showing you the neuronal subclusters in T-SNE space, which included about 20 distinct clusters of excitatory and inhibitory neurons. And I'll just put in a plug that we've also done single nucleus RNA-seq and 4 other regions of the human brain important for reward processing, the nucleus accumbens, the amygdala, hippocampus, and the subgenual anterior cingulate cortex. And you can find all these data on bioRxiv if you're interested in exploring further. But back to the spatial registration, when we correlate the enrichment statistics from these DLPFC subclusters with those from our laminar Visium data, we find that specific subclusters of excitatory neurons correlate with either deeper superficial cortical layers. So for example, you can see excitatory neuron clusters 13, 10, and 27 likely represent layer 6 pyramidal neurons. We also see that oligodendrocyte clusters are enriched in the white matter, and inhibitory interneuron clusters are scattered throughout the cortex as expected. In addition to our own data, we've also done spatial registration for other publicly available cortical data sets to add spatial context to existing single cell data. So for example, we did this for single nucleus RNA-seq data from 48 donors across 71,000 nuclei obtained from the human prefrontal cortex in a study of Alzheimer's disease by Matthews and colleagues. So the heat map shows the correlation between the enrichment statistics computed on the single nucleus RNA-seq data on the x-axis against those from our laminar Visium data on the y-axis. So we were able to assign different neuronal clusters to specific cortical layers. And interestingly, our analysis showed that the excitatory neuron subclasses 2, 4, and 6, which were most associated with clinical traits of Alzheimer's disease, were preferentially localized to the upper layers, or layers 2, 3 of the DLPFC in our data. So analyses like these can help us to better understand the function of particular cell types residing in specific anatomical locations or subdomains. In terms of data integration, one new tool called Tangram from the Regiv lab that we're particularly excited about actually aligns single nucleus RNA-seq data to various forms of spatial data, including Visium data, collected from the same brain regions, as well as to histological images and to public atlases. So Tangram is equipped with a deep learning computer vision pipeline, which allows for automatic identification of anatomical annotations on histological images. So here in a test case, we use Tangram to map single nucleus RNA-seq data from the DLPFC onto a Visium DLPFC tissue section. So you can see that the predicted location of dissociated single nuclei, previously annotated as excitatory neurons belonging to layers 4 and 5, matches really nicely with expression of PCP4, a gene highly expressed in cortical layers 4 and 5. Tangram also provides opportunities for performing single cell deconvolution of spots, where in the mouse cortex, the authors demonstrate mapping of different cell types onto single cells segmented within individual Visium spots. So currently these assignments are random to a selected segmented cell within the spot, but there are of course opportunities to try to pair cell types to known morphologies. So for example, excitatory neurons have a pyramidal shape that could be leveraged to get more precise cell assignments. Integrating these maps with anatomical atlases and a common coordinate framework will also be a high priority for the field. So another area of focus for our group and the spatial transcriptomics field in general has been to develop data-driven unsupervised approaches for annotating and clustering Visium data. So everything I've been discussing so far in the DLPFC has been in terms of this supervised pseudo-bulking approach. But of course, for regions where the spatial organization is less well known, or instances where you want to discover non-laminar patterns of spatial gene expression, data-driven approaches are required. So in collaboration with Stephanie Hicks and Lucas Weber at Johns Hopkins, we employed several implementations of unsupervised clustering methods with spot level Visium data using spatially or highly variable gene sets. Our best implementation was a sort of semi-supervised approach, which involved unsupervised clustering guided by the layer-enriched genes identified using our differential expression enrichment models. While we did try actually incorporating the spatial coordinates of spots into our clustering model, and we did see a small improvement, our approach did not actually take into account any of the histology data. However, since we made our data publicly available upon preprint, several other groups were able to develop new methods for data-driven clustering, and I'd like to mention a few of them here that were recently published or are currently in preprint. So the first method is BaseSpace, which is a fully Bayesian spatial clustering method that models a low-dimensional representation of the gene expression matrix and encourages neighboring spots to belong to the same cluster via a spatial prior. So here you can see BaseSpace was able to outperform other clustering methods to nicely resolve cortical layers in our DLPFC data. Again, here though, this method focuses entirely on gene expression and does not actually leverage information from the histological data. However, a tool from the Lee lab called SPA-GCN has focused on integrating gene expression, spatial location, and histology to identify spatial domains as well as spatially variable genes. So SPA-GCN starts from integrating gene expression, spatial location, and histology information using a graph convolutional network, then separates spots into different spatial domains using unsupervised iterative clustering. So the graph convolutional network is based on an undirected weighted graph in which the edge weight between every two spots is determined by Euclidean distance between the two spots, defined by the spatial coordinates xy, and then a third dimensional coordinate z obtained from the RGB values in the histology image. So you can see in this example in mouse brain that SPA-GCN outperforms other clustering methods to identify biologically meaningful spatial domains and identify the most robust marker genes for these domains. The authors have also applied this tool to our DLPFC data, and you can find that example and more information on how their tool works in their preprint that's on bioRxiv. For those of you who might be interested in working with spatial transcriptomics data, I want to plug two tools that our group and collaborators have been involved in developing that might be of interest for working with these kinds of data sets. The first is Spatial Experiment, which is infrastructure for spatially resolved transcriptomics data in R using bioconductor that both Lucas Weber and Leo Collado-Torres have been involved in developing. So Spatial Experiment extends the existing single cell experiment for single cell data from the bioconductor framework, which basically brings with it advantages of modularity, interoperability, standardized operations, and comprehensive documentation. And the idea here was really to improve efficient analysis of spatial data to facilitate storage, retrieval, subsetting, and interfacing with downstream tools. If you're interested in exploring our data or working with tools to explore other spatial transcriptomic data sets of your own, I'd also encourage you to check out our preprint Spatial LABD, which describes an R bioconductor package to interactively explore spatially resolved transcriptomics data generated with the 10x Genomics Visium platform. The package contains functions to interactively access, visualize, and inspect the observed spatial gene expression data and data-driven clusters identified with either supervised or unsupervised analysis, either on the user's computer or through a web application. So in the last part of my talk, I just want to touch base on how we are using spatial transcriptomics to better understand complex brain disorders, including psychiatric disorders, as well as neurodegenerative disease. So to mention one highlight from our DLPFC study, we wanted to ask if different neurodevelopmental and neuropsychiatric gene sets were enriched in particular cortical layers. So this is a heat map showing enrichment of different autism spectrum associated gene sets on the x-axis and across the cortical layers on the y-axis. So we found that ASD associated genes have strong odds ratios for layers 2, 5, and 6, And we confirmed the layer 2 and 5 associations using data from a recent exome sequencing study, which identified about 102 genes with ASD-associated variants. And interestingly, stratifying these genes by their clinical symptoms refined the laminar enrichments. So the 53 genes associated with ASD dominant traits were more enriched for layer 5, whereas the 49 genes associated with neurodevelopmental delay were more enriched for layer 2. So this could suggest that different functional subclasses of neurons might be potentially contributing to each clinical subgroup. I'll also just mention that all these data are available for exploration at our Shiny app, and you can also upload your own clinical gene sets of interest. So beyond the clinical relevance of our initial DLPFC study in neurotypical control subjects, we're also working to directly understand complex brain disorders. So to do this, we've been setting up a new application of Visium called Visium immunofluorescence, or Visium-IF. So this flavor of Visium is particularly exciting because it allows for integration of proteomic and transcriptomic data. So the workflow is exactly the same as I described before, but instead of doing H&E histology, we use fluorescence antibody staining for proteins that label different cell types, subcellular compartments, or even neuropathology. So this immunofluorescence map is then directly integrated with transcriptomic data, enabling us to analyze relationships between gene and protein expression within the same tissue slice. So to give you a sense of one of our antibody panels, here's an example of immunostaining for 4 of the main cell types in the human inferior temporal cortex, NUN labeling neurons in green, TMEM119 labeling microglia in white, GFAP labeling astrocytes in yellow, and OLIC2 labeling oligodendrocytes in pink. So using this approach, we can now start to understand the cell types and the cellular compartments that contribute to the gene expression profile in each of these Visium spots. So we're currently employing Visium-IF in the human cortex to understand molecular and cellular changes associated with pathology in neurodegenerative disease. So in Alzheimer's disease, we know extracellular amyloid beta plaques and intraneuronal neurofibrillary tangles containing hyperphosphorylated tau are key pathological hallmarks. These plaques and tangles accumulate in the brain during progressive neurodegeneration, and they're associated with disease severity. So here's an example of immunostaining for A beta in green, phospho-tau in white, and a dendrite marker MAP2 in red. And this is from a tissue section of inferior temporal cortex from a donor with late-stage Alzheimer's disease and severe pathology. I'll also just point out that the size of the plaques is roughly equivalent to the size of one of the Visium spots, about 50 microns. We've just completed a Visium experiment using this antibody panel, and we're currently working on analyzing our sequencing data and integrating it with these protein maps. And again, this is an excellent opportunity for computer vision approaches in the analysis of this rich immunofluorescence data, particularly how to quantify different features of pathology that can be integrated with the corresponding gene expression data. So just to summarize, as I've been discussing our kind of current and ongoing spatial transcriptomics work, I've really tried to highlight how we can leverage the microscopy data generated in the Visium assay for integration with gene expression data. I've listed out a few of these ideas here, including improved mapping of single cells or cell types onto histological images for high resolution molecular atlases, linking gene expression to different cellular compartments like neuropil, axons, and dendrites, understanding local gene expression in the context of pathology and Alzheimer's disease, discovering new morphological features associated with changes in gene expression, which is particularly relevant for brain disorders without obvious pathology. And lastly, improving data-driven clustering to identify previously unknown spatial topography. So as spatial transcriptomic assays continue to increase in resolution and multiplexing, and they begin to include more multi-omics data, I think you'll see more and more opportunities for incorporating the corresponding microscopy data. I think the field is really just getting started with this. And if you're interested to learn more about our work or exploring these kinds of data, you can find at the link below at research.labd.org/spatiallabd. And with that, I'll acknowledge our spatial team, especially my collaborators, Carrie Martinovich, Leo Collado-Torres, and Stephanie Hicks. We have some exciting projects on the horizon and lots more Visium data to come. So I'm happy to try and answer your questions as best I can, and I'd really love to hear your ideas on how we can sort of better harness our microscopy data. We're always looking for collaborators with expertise in image analysis who are interested in working with these novel kinds of data sets. So thanks for your attention.
Thanks so much, Kristen. That was great. And I appreciate your sensitivity to the audience and your penultimate slide sort of listing the path. What are the next steps that one can take to be part of this this kind of amazing new application? Maybe I could just start with a quick question that's just sort of technical and straightforward, but does address one of the current limitations, and it's really number one on this list. So when you describe a spot, and you said it's about 50 microns in diameter, as you know, in terms of nuclei size, that's somewhere between 1 and 10 nuclei, depending on what the packing density is. So in this presentation and in other presentations earlier, there's clearly a benefit in grouping and averaging. But one of the issues in the nervous system is that there's incredible local cell diversity, and when one defines a spot as a certain cell type, or it has a very high signal from a certain cell type, of course that's the mean signal is sort of dominated by one cell type, but of course there are neighbors that are influencing that. So the first part of that is how do you deal with competing expression within one spot? How do you define a spot as being dominated by one cell type? And I guess the second part is what path do you see in going from that group average to a higher resolution analysis of individual gene expression?
Yeah, that's a great question, very relevant, and something I think that people are grappling with as new developments come to the technology. So right now, in terms of computational tools, there's also quite a lot of focus on this deconvolution approach. So how you can try and break that spot out into smaller spots, or using the histology data, know how many cells are in there based on the morphology, maybe try and correlate that with whether it's an excitatory neuron or inhibitory neuron. So I think for data that is where a spot includes many cells, deconvolution has been what people are interested in. However, I think you'll see more and more, and especially even from 10x Genomics, that they're now appreciating people want higher single cell resolution. So they're making their spots even smaller. And there, it's the opposite challenge. So now with some of the new HD platforms that are coming out, you will likely have multiple spots per cell. So you will have the opposite problem where you need to put your spots together, right, to kind of build one cell. But I think the higher resolution for some of the unsupervised approaches is probably better, because you may be able to associate some of these very small spots that might only be 5 microns with different features of a neuron, like perhaps a process or a cell body. So both sides, high resolution and then low resolution, have their own problems, I guess.
Maybe time for one more question to kind of keep on schedule.
I was curious about how the morphology of the cells and protein expression kind of occurs at what I would call a dramatically different time scale than RNA expression. And then so trying to cross-correlate those two or co-correlate those two could lead to kind of spurious associations of morphology or protein expression, especially for the fluorescent work that you showed, and trying to decouple those or trying to figure out how those are related. What kind of work goes on there, or how do you try to disentangle those two?
Yeah, I mean, that's a good question. So right, by the time you have some kind of pathology, right, you already have probably pretty dramatic changes in RNA expression. So, one particularly interesting application of something like, the Visium-IF is can you do this earlier, right, before you actually have a pathological burden, and start to predict who may or may not progress to that kind of burden. But I think because it's only become so new that we can do the molecular profiling and the pathological profiling at a genome-wide kind of scale, I think like that remains to be seen. I think some of these challenges.
Great talk.
Let me close that. Thanks so much. And one of the deficits of a Zoom-based meeting is that we don't really get to interact with each other. But if we were, you'd realize that Kristen is about to have a baby, and I'm glad that we got to this point before she comes. But good luck with that next week. I know that will be a big part of your life, and I hope that there are lots of Legos that will make her happy going forward.
Yes, well, I'm glad she did not arrive before before I had the opportunity to meet you all.
Great, thanks, Kristen. So I'll hand it back, I guess, to May and Dimitri for the next session.
Yeah, thank you, Dan. So and thanks, Kristen, for an excellent talk. So the next speaker is Keith Chang, who is a very well-known professor at Penn State University. He's going to talk to us about the geometry of life, micro-CT as a foundation for computational organismal and tissue phenomics. Just to give you a little bit about his background, he's interested in making fundamental contributions to the understanding of genetic and molecular mechanisms involved in human biology and disease. His lab specializes in genetic aspects of human disease, use of model systems such as zebrafish, and he has done contributions to web-based scientific resources and potentially high throughput forms of 3D imaging. Some of his specific work is aimed at increasing understanding on the basis of phenotypic variability, particular as it may impact cancer. Also, he's interested in basic mechanisms underlying the relation between human skin pigmentation and skin cancer, contributing to web-based infrastructure for science, education, and public service, and working towards 3D derivative of histology that also allows the identification and characterization of cell types, utilizing computers basically define slice angle, thickness, and other important variables. So